Dear students, welcome to the class on virus structure and replication. As we know, viruses are the smallest known infective agents and they are the simplest form of life known. They do not possess any cellular organization and they do not fall into the category of unicellular microorganisms. Coming to the properties of viruses, viruses do not have a cellular organization. They contain only one type of nucleic acid, either DNA or RNA, but never both type of nucleic acid. Viruses are obligate intracellular parasites. They lack the enzymes necessary for metabolism, for protein and nucleic acid synthesis, and thus they are dependent for replication on the synthetic machinery of the host cell. As a result, they are obligate intracellular parasites. Coming to the morphology of a virus, regarding their size, as we know, viruses are very smaller than bacteria, and as a result, they could pass through bacteriological filters. And this nature, this filterability, their ability to pass through bacteriological filters lead to their recognition, lead to their discovery as a type of infectious agents. Due to this nature, upon discovery, they were termed as filterable agents. The size of a virus vary widely in size from 20 to 300 nanometer. The largest virus is a pox virus having about 300 nanometer size, and it is as large as the smallest bacteria. The smallest viruses belong to the parvovirus group and their size is approximately 20 nanometer. The viruses are very small to be seen under a light microscope. So they could be observed only by using an electron microscope. Some larger viruses, such as the pox virus, can be observed under a light microscope when it is suitably stained. All other viruses are visible only when observed under an electron microscope. Shape of a virus. The shape of a virus varies with different groups of viruses. Most of the animal viruses are roughly spherical. Some are irregular and pleomorphic. Pox viruses are brick-shaped. A rabies virus is bullet-shaped. A tobacco mosaic virus is rod-shaped. Bacteriophages, the viruses that attack bacteria, are having a complex morphology. The structure of a virus. A virus consists of a nucleic acid core surrounded by a protein coat. This protein coat is known as capsid, and this nucleic acid structure surrounded by this capsid together is known as a nucleocapsid. So essentially each virus is a nucleocapsid. This capsid is composed of a large number of small units known as capsomeres. Capsomeres are polypeptide molecules which are arranged in a symmetrical fashion. So this capsid with the enclosed nucleic acid is termed as a nucleocapsid. There are three types of capsids based on their arrangement of capsomeres. They are icosahedral symmetry, helical symmetry, and complex symmetry. So, let us discuss about the various types of symmetries. They are icosahedral symmetry, helical symmetry, and a complex structure or a complex symmetry. So, what is an icosahedra? Icosa means 20 in Greek. An icosahedra is a structure, is a polygon with 12 vertices or corners and 20 facets or sides. Each facet, each side in an icosahedra is in the shape of an equilateral triangle. Two types of capsomeres are generally present in an icosahedral capsid. They are the pendants and the hexans. Adenovirus is an example having an icosahedral symmetry. The next type of asymmetry present in virus, the next type of structure present in a virus is helical symmetry. Here, the nucleic acid and the capsomeres are wound together in the form of a helix or spiral. Example is rabies virus. And the third type is a complex symmetry. These types of viruses do not have either icosahedral or helical symmetry due to the complexity of their capsid structure. An example is pox virus. And some viruses having this complex symmetry, for example, bacteriophages such as T4 phage, they have both an icosahedral and a helical structure. In the case of T4 phage, it have an icosahedral head and a helical tail, and thus it is said to have a complex symmetry. We can see different types of structures, different types of viral symmetry. The icosahedral symmetry is here. The helical symmetry, pox virus having a brick shape and a bacteriophage having a complex symmetry. Both pox virus and a bacteriophage are having a complex symmetry. So, what are the functions of a capsid? A capsid protects the viral genome from physical destruction and enzymatic inactivation from various environmental factors. A capsid provides binding site for the virus to attach to specific receptor sites on the host cell. Capsid facilitates the assembly and packaging of virus after replication. It serves as a vehicle of transmission from one host to another. A capsid is antigenic and it is specific for each virus type, and as a result, it is of importance in the host defense to virus infection as well as for the preparation of vaccines. A capsid provides a structural symmetry to the virus particle. So these are the functions of a capsid. So in the structure of a virus, the viruses are essentially a nucleocapsid. They are nucleic acid surrounded by a protein capsid. So a virus having only the nucleocapsid structure are non-enveloped or naked viruses. There are certain other viruses which are enveloped, having an outer envelope around this nucleocapsid structure. So in an enveloped virus, the envelope or the outer covering of the virus is derived from the plasma membrane of the host cell during the virus replication. The envelope is generally lipoprotein in nature, and the lipid is also derived from the host cell. The enveloped viruses are susceptible to the action of lipid solvents. Since their outer envelope is generally mainly made up of lipids, they are highly sensitive to lipid solvents such as ether, chloroform, detergents, alcohol, etc. While the viruses which are non-enveloped or naked viruses which are having only the nucleocapsid structure are resistant to the lipid solvents. So enveloped viruses are having an outer envelope or outer structure external to the nucleocapsid, and these viruses are generally susceptible to or sensitive to the action of lipid solvents. In enveloped viruses, they have an outer projection or a glycoprotein spikes on the outer surface of the envelope. And these outer spikes or glycoprotein spikes are known as peplomers. Peplom means envelope, and peplomers are projecting spikes on the outer surface of an envelope in an enveloped virus. A virus may have more than one type of peplomer. For example, the influenza virus have two types of peplomers. The hemagglutinin spike, which is triangular in shape, and the neuraminidase spike, which is mushroom shaped in structure. So, the peplomers and the envelope provides a chemical antigenic and biological properties to the virus. So let us see what are the functions of a peplomer. Many peplomers mediate attachment of the virus to the host cell receptors to initiate the entrance of the virion into the cell. Some viral glycoproteins attach to receptors on the surface of red blood cells and cause the cells to agglutinate, and this property is known as hemagglutination. Some peplomers possess enzymatic activity like neuraminidase, which cleave the neuraminic acid on the surface of host cell glycoproteins. And generally, the peplomers are major antigens. Coming to the major part in a virus structure, their nucleic acid. As we already discussed, a virus will have either DNA or RNA as its genetic material. Virus contain a single kind of nucleic acid, either DNA or RNA, and this particular nucleic acid will encode the genetic information necessary for the survival and replication of virus. The genome may be single-stranded or double-stranded. It may be circular or linear, and it may be segmented or non-segmented. The type, structure, and size of a nucleic acid, of a viral nucleic acid, is used for the classification of viruses. So, a viral genome may be either a DNA or an RNA. It may be either linear in structure or it may be circular. It may be either single-stranded or double-stranded. An example for single-stranded linear DNA virus is parvovirus. Double-stranded linear DNA virus is herpes virus. Single-stranded circular DNA virus is M13. Double-stranded circular DNA virus is baculovirus. Single-stranded linear RNA virus, tuberculous virus. Reovirus is a double-stranded linear RNA virus, and hepatitis D virus is a single-stranded circular RNA virus. Now, we will discuss about the procedure or the process of viral replication. The genetic information necessary for viral replication is in the viral nucleic acid, but as we said earlier, a virus lacks the biosynthetic enzymes necessary for replication. So, they depend on the host cell for their replication. The viral replication or viral multiplication cycle is divided into 6 phases or 6 stages. They are adsorption or attachment, penetration, uncoating, biosynthesis or replication, assembly or maturation, and release. These are the different phases in a viral replication cycle. So, coming to the first stage in viral replication, that is adsorption or attachment. So, when a virus comes into contact with a sensitive host cell, a susceptible host cell, it is generally by random collision. And after this random collision, it will be followed by specific adsorption or attachment. And the specific adsorption or attachment is mediated by the binding of virion surface structures known as ligands, which are present on the surface of the virus, which will bind to the receptors which are present on the host cell surface. In the case of influenza virus, the hemagglutinins spike or the hemagglutinins peplomer will bind specifically to the surface of the respiratory epithelium, host respiratory cells, on their sialic acid residue, which is present. The sialic acid residue is present on the surface of the host cell, generally the cells in the respiratory epithelium. While in the case of human immunodeficiency virus, the virus surface glycoprotein GP120 will act as a ligand which will bind to the mature T lymphocytes. They will bind to the CD4 glycoprotein which is present on the surface of the T lymphocytes. If the virus is a rabies virus, it will bind to the neuronal cells on the acetylcholine receptors. So, during adsorption or attachment, a virus will specifically bind to a sensitive host cell by using their surface ligands. The ligands are present on the virus surface, and this ligand will bind specifically to receptors which are present on the host cell. After adsorption, the next stage in a viral replication cycle is penetration. After adsorption, the virus particle is taken up inside the cell. There are different mechanisms for penetration. Some viruses accomplish penetration or they get entered into the host cell by receptor-mediated endocytosis or viropexis. Here, uptake of the ingested virus particle occurs by the use of an endosome. This is the mechanism by which most non-enveloped viruses gain entry or they get penetrated into the cell. This process is known as viropexis. In the case of enveloped viruses, the virus will fuse their outer envelope, the outer membrane, with a host cell plasma membrane and they deliver their nucleocapsid or the genome directly into the host cytoplasm. So, naked viruses employ viropexis, where it is a modified endocytosis. The viruses are uptaken by endosomes, while in the case of enveloped viruses, the virus envelope will fuse with the host cell membrane. After this, the nucleocapsid or the nucleic acid will get transported directly into the host cytoplasm. After penetration, the next stage in a virus replication is uncoating. Uncoating is a stripping of the virus of its capsid or the removal of the capsid so that the nucleic acid is released into the cell for its replication. Uncoating is generally carried out by host cell lysosomal enzymes. Genome of most RNA viruses will remain in the cytoplasm where it will get replicated, while the genome of DNA viruses are delivered into the nucleus. The next phase in the viral replication cycle is biosynthesis or replication. This phase includes the biosynthesis of viral nucleic acids. Capsid protein will be produced and the enzymes necessary for the various stages of viral synthesis, assembly and release are also made during this phase. In addition to these molecules such as the viral nucleic acid, capsid proteins and enzymes, some regulatory proteins are also synthesized during this phase, and these regulatory proteins are required to shut down the normal cellular metabolism of the host cell so that the host cell machinery is completely directed towards the production of virus components. Most DNA viruses synthesize their nucleic acid in the nucleus of a host cell. There is an exemption, that is pox virus. Pox virus will synthesize the components in the host cell cytoplasm, not in the nucleus. Most RNA viruses synthesize their components in the cytoplasm. Here also there are some exemptions. Orthomyxovirus, paramyxovirus and retrovirus will synthesize few of their components in the nucleus. All the viral proteins are synthesized in the cytoplasm. So there are various stages in the biosynthesis stage. They are the transcription of mRNA, messenger RNA from viral nucleic acid, translation of messenger RNA into early proteins, replication of viral nucleic acid, and the synthesis of late proteins. So, during the first stage, transcription of messenger RNA or mRNA will be done by using the viral nucleic acid. The viral nucleic acid may be either a DNA or an RNA. From the viral nucleic acid, mRNA will be transcribed. After this primary transcription, the mRNA synthesized during the first stage, first transcription, will be translated into early proteins by using the host cell machinery. These early proteins are enzymes required for the synthesis of virus components, and also they will induce shutdown of the host machinery. They will inhibit the host protein and host nucleic acid synthesis. So the early proteins are synthesized soon after viral infection. They will carry out their function include to replicate virus nucleic acid. They will shut down the host machinery. Generally, these early proteins are enzymes. They are generally catalytic. And the early proteins are synthesized only in very small amounts. After the translational synthesis of early proteins, the third stage is a replication of viral nucleic acid. The viral nucleic acid will be replicated to several copies. After replication of viral nucleic acid, the late proteins are synthesized. The late proteins are also known as structural proteins, and they are the components of daughter virus capsids. So, the later proteins are synthesized later after some time after the viral infection. These late proteins are the proteins required for virus coat or capsid. They are more structural. They do not have much of catalytic activity. They are the structural components. And when compared to early proteins, the late proteins are synthesized in larger amounts. So after the biosynthesis stage, the next stage in a viral replication cycle is assembly or maturation. Assembly of the various viral components, the capsids and the nucleic acid. They will be getting assembled to form the virions or new viruses during this phase, during the assembly or maturation phase. This may either take place in the nucleus or in the cytoplasm. Herpes virus and adenoviruses are getting assembled in the nucleus, while picornaviruses and pox viruses are examples of viruses which get assembled in the cytoplasm. After assembly, in the case of enveloped viruses, they need an outer envelope also. All other viruses have the nucleic acid and the capsid, but the enveloped viruses will have an outer envelope also. So they derive their envelope from the host cell nuclear membrane or host cell plasma membrane. Herpes virus is an enveloped virus which derived its envelope from the host nuclear membrane, while orthomyxoviruses and paramyxoviruses are examples of viruses, enveloped viruses, which derive their envelope from the host cell plasma membrane. So, the nucleic acids and the capsids will be assembled to form new virus particles. We can see the assembly of a helical capsid, icosahedral capsid, etc. in this image. According to the type of the virus, the capsids will arrange themselves around the viral nucleic acid to form a complete virus. And after the assembly, the last phase in a viral replication cycle is release. The release of the progeny virus particles from the host cell into the environment. Viruses are released from the host cells, after lysis of the cell, or by exocytosis, or by budding from the plasma membrane. The viruses may either lyse the cell, or they can come out of the cell by exocytosis, or some viruses bud out of the plasma membrane into the exterior. Viruses, for example, polioviruses, which have a nucleocapsid only in their structure, they are released by the lysis of the host cell. Or some viruses may get extruded, may get transported to the outside of the cell by a process known as reverse phagocytosis, which can be observed from the first image. The release of the enveloped viruses, it occurs as a bud. It occurs by budding from the plasma membrane, and the budding is the mechanism by which enveloped viruses get released from the host cell, And as a result, since they get released through budding, there is no killing of the cell during this release. The enveloped viruses get released as buds through the plasma membrane of the host cell. So, in a viral replication, the first phase is adsorption, during which the virus particle get attached onto the surface of a sensitive host cell, and after adsorption or attachment, the virus will get penetrated into the cell. After penetration, the virus will undergo uncoating so that the nucleic acid get released. The next phase is the biosynthesis or replication, during which there will be transcription for the synthesis of early proteins, replication of the virus nucleic acid, and the production of late proteins which are necessary as the capsid proteins. And after this biosynthesis phase, the next phase is the assembly, during which the replicated nucleic acids will get assembled by the proteins, capsid proteins, to form new virus particles. And after assembly and maturation, the virus particles will get released from the cell, either by the lysis of the cell, or by exocytosis or reverse phagocytosis, or by budding from the plasma membrane of the host cell. So these are the various phases in a viral replication. Based upon the replication mechanisms, the viruses are classified into 6 classes by Baltimore. So, the Baltimore classification of viruses is based on the replication mechanism. The 6 classes of virus based on Baltimore classification are single-stranded DNA viruses, double-stranded DNA viruses, single-stranded RNA viruses having positive strand, single-stranded RNA viruses having a negative strand, double-stranded RNA viruses, and retroviruses. The first class is a single-stranded DNA virus. These viruses have their nucleic acid a single-stranded DNA. The single-stranded DNA molecule will move into the host cell nucleus during their replication and will be converted into double-stranded form. Transcription is achieved by host enzymes, and an example for single-stranded DNA virus is a parvovirus. The second class of viruses as per the Baltimore classification is double-stranded DNA viruses. They have a double-stranded DNA as their genome, and this double-stranded DNA enters the host cell nucleus, and they also use the host cell enzymes for their transcription. Examples are heparina viruses. Baltimore classification class 3 is single-stranded RNA viruses, and these single-stranded RNA viruses are classified into 2 categories, class 3 and class 4, depending on the mechanism or the method of mRNA transcription. The first one is, or the third class is, single-stranded RNA virus having a positive strand or plus strand or a positive sense strand. Here, the viral RNA directly can act as the mRNA or the messenger RNA. The viral RNA is infectious by itself, and it is translated directly into viral proteins in the host cell cytoplasm. Examples are picornavirus, toga virus, etc. The fourth class, as per the Baltimore classification, is a single-stranded RNA virus, where the RNA is of negative sense, negative strand or minus sense. Here the RNA is antisense. It cannot act as an mRNA. The virus possesses their own RNA polymerase in the virus, which will synthesize the messenger RNA from the viral genome. So, the viral RNA is antisense, having a polarity opposite to the mRNA. It will act as a template for the synthesis of mRNA, and the enzymes for this synthesis is already present in the virus. The virus have their own RNA polymerase for the synthesis of mRNA. Examples are rhabdovirus, orthomyxovirus, paramyxovirus, etc. The fifth class of Baltimore classification of viruses is double-stranded RNA viruses. Here, the genetic material is a double-stranded RNA which is transcribed to form a messenger RNA or mRNA, and this is carried out by viral polymerases, viral RNA polymerases. Example is a reovirus. And the last and sixth class of virus is retrovirus. Here, the virus is having a single-stranded RNA genome which will be converted into an RNA-DNA hybrid by a special enzyme known as a reverse transcriptase enzyme. This reverse transcriptase enzyme is a viral enzyme which is already present in the virus. From the DNA-RNA hybrid, a double-stranded DNA is then synthesized, and this double-stranded DNA is known as provirus. It can integrate itself into the host cell chromosome, and it can be maintained in such provirus form for a long period of time. And this integration of the virus into the host cell chromosome to form the provirus may lead to the transformation of the host cell, and this generally results in the development of neoplasia or cancer in the host cell. The retrovirus integration into the host chromosome as a provirus may cause the transformation of the cell to form a cancerous cell. So, we were discussing about viruses, their shapes, different structural symmetries, different stages in their replication, and we also discussed about the different classes of viruses based on their replication mechanism. Thank you so much for listening.